CSIR Evolution, Chapter 11. Chapter 11, Innate and Learned Behavior. Animal behavior includes all the ways animals interact with other members of their species, with organisms of other species, and with their environment. Behavior can also be defined more narrowly as a change in the activity of an organism in response to a stimulus, an external or internal cue or combination of cues. Behavior can be categorized into two main types: innate or inherent behavior, or learned or acquired behavior. 11.1 Innate or inherited behavior. Much of the innate behavior of many animals is highly stereotypic. It is performed in the same way every time, and species-specific. There is little variation in the way different individuals of the same species perform it. We can identify species of spiders by their specific web designs. Web spinning requires thousands of movements performed in just the right sequence, and for a given species, most of that sequence is performed the same way every time. Different spider species spin webs of different designs, using different sequences of movements. Thus, innate behavior or stereotyped behavior is an inborn or inherent behavior sequence of activities which is predictable, species-specific, genetically controlled and independent of past experience. Since this type of behavior follows a fixed action pattern, FAP, which is predictable and found in all members of a species, it also called stereotyped behavior. 11.2 Characteristics of innate or stereotyped behavior. Pattern of behavior is inherited, that is, it is passed on from parents to offsprings. It is unlearned behavior. It occurs in all the members of a species, hence it is species-specific and predictable. It is not dependent on past experience as it is an inborn response to a stimulus. It takes place even when kept in isolation away from their fellow members. Innate behavior has high adaptability and survival value. Web spinning by spiders is an example of a complex behavior that requires no learning or prior experience. In many species of spiders, when juvenile spiders hatch, their mother is already dead, and they disperse immediately. They have no experience of their mother's web. Yet when they construct their own webs, they do it perfectly, without the benefit of experience or a model to copy. In fact, their web-spinning behavior is resistant to modification by learning. When confronted experimentally with obstacles to web construction, young spiders appear incapable of learning how to modify the design of their webs. 1.3 Types of innate behavior. Innate behavior is behavior that's genetically hardwired in an organism and can be performed in response to a cue without prior experience. It is of the following types. 1.3.1 Reflexes. A simple movement of a part of the animal in response to a stimulus is called reflex. It is a quick, innate, and immediate response of a part of the body to an external or internal stimulus, which has great adaptive and survival value to the organism. Reflexes are inherited and unlearned behavior found in all members of the species. The knee-jerk, constrictive pupil of eye in the bright light, blinking of eye, peristalsis, coughing etc. in man, flight in birds, web spinning in spiders are all examples of reflexes. A reflex action requires a reflex arc which consists of a sensory organ, receptor, a sensory nerve, afferent nerve, the spinal cord or brain, an intermediate motor or efferent nerve and a motor organ or effector. A reflex action thus involves the sense organs and nervous system. The stimulus is received by the receptor or sense organ which sets up a sensory impulse. This impulse is transmitted to the central nervous system, brain and spinal cord, through the intermediate nerve. This motor impulse is transmitted to the effector or motor organ, muscles, and glands, which either contract or secrete in response to the stimulus. Many patterns of animal behavior are complex combinations of simple reflex. Advantages of reflex: it enables the animal to respond immediately to harmful stimuli, hence it has great adaptive and survival value. Since Many of the reflex actions are controlled by the spinal cord. It relieves the brain from too much work. 1.3.2 Instinct. Instinct is the most complex type of stereotyped behavior, which is unlearned, predictable, genetically controlled and species-specific, and it is in response of a sign or release or stimuli. Releasers are usually a specific component of the sensory information available to an animal. Adult. Male European robins, for example, have red feathers on their breasts, which serve as releasers of aggressive behavior in other males. During the breeding season, the sight of an adult male robin stimulates another male robin to sing, perform aggressive displays, and attack the intruder if he does not heed these warnings and retreat. An immature male robin, whose feathers are all brown, does not elicit this aggressive behavior. A tuft of red feathers on a stick, however, is a sufficient releaser for male aggressive behavior, even though it looks nothing like a real robin. Of all the stereotyped behavior, instincts are the most fascinating to study. E.g., building of nest by birds, singing to attract males, territoriality, migration, parental care, etc. 1. Characteristics of instinctive behavior. 1. Instinctive behavior is in response of a sign or release or stimulus. Sign stimulus is one which triggers an instructive response, which could be a part of the whole stimulus. A release or stimulus is one which triggers a series of stereotyped behavior. 2. It is the result of both external stimuli and inner environmental conditions. External stimuli represent environmental factors like temperature, photoperiods, suitable nesting material, food sources. The internal conditions refer to the level of hormones in the blood. 3. Though it is categorized as unlearned and stereotyped behavior, it can be modified by exposure, learning, and experience. E.g., a male bird will sing a simple song when kept in isolation. Birds need to hear the songs of adults to be able to sing the complete song. 4. External stimuli are able to trigger a series of instinctive behavior, provided the internal conditions are conducive. If there is sufficient concentration of hormones in the bird and minimum external stimulus is sufficient to trigger the entire series of behavior. 2. Examples of instinctive behavior. Example 1. Egg rolling in waterfowl, European greylag goose. When an egg is removed from the nest of an incubating goose and placed a few inches in front of her, she will reach out with her neck and pull the egg back into the nest, figure 91a. While perhaps appearing rather insignificant to the casual observer, this behavior suggested a number of important ethological concepts to Lorenz and Tinbergen, who carefully studied its characteristics in the European greylag goose. After it was confirmed as innate by observing it in inexperienced geese, egg rolling was analyzed in terms of its behavioral components. It was noted that if, while the goose was rolling the egg back toward the nest, the egg veered off to the side, the goose steered it back toward the nest. This taxis component, which steers the behavior relative to the environment. We may ask whether egg rolling is the same sort of behavior a golfer exhibits when he or she reaches out with a golf club to retrieve a ball that has rolled into a ditch. Human behaviors of this type volitional behaviors are based on conscious choice or decision. Although the mental states of animals are not directly accessible to us, one component of egg rolling behavior sets it apart from most human volitional behavior. If the egg is snatched away from the goose while she is retrieving it, she continues the retrieving movement even though there is no longer an egg to retrieve. Thus, once begun, the behavior is relatively independent of the external stimulus that initiated it. This characteristic is certainly not typical of human volitional behavior, although it superficially resembles certain compulsive behaviors in humans. Lorenz named this type of behavior, which seems to be neither a simple reflex nor a volitional behavior, a fixed action pattern, FAP. Although the behavior is not fixed in a literal sense, this term emphasizes that it is more or less invariable, suggesting a strong genetic component. Any stimulus that elicits a FAP such as the egg and egg rolling behavior or the red-colored belly in the attack behavior of the stickleback fish is called a sign stimulus. For example, a goose will retrieve a wooden egg in the same way as a real egg, so the wooden egg is also by definition a sign stimulus. Yet only the real egg is the natural sign stimulus, that is, the one that evolved with the behavior. The special term releaser was originally applied only to the latter category of sign stimuli. Curiously, artificial sign stimuli, such as wooden eggs, are sometimes more powerful than releasers. Certain birds, figure 91b, when given a choice, will choose a large wooden egg in preference to their own normal eggs. In this case, the stimulus has been called a super sign stimulus. Sign stimulus are detected in central nervous system by special sensory mechanisms, called innate releasing mechanisms, IRM. Each IRM is specific for particular sign stimulus, thus oval-shaped eggs, whether real or artificial, are detected by IRM for this shape. The IRM then sets into motion FAP, egg rolling in this example. It was observed that additivity of independent and heterogeneous features of a stimulus positively affects and elicit more frequency behavior. This is referred as the law of heterogeneous summation. For example, the herring gull recognizes its eggs by various features, including size, shape, background color, and speckling. These features have been shown to be additive in their effects upon retrieval of eggs that have rolled out of the nest. Figure 91, innate behavior, A, egg rolling behavior, B, super sign stimulus. Example 2. The reproductive behavior in three-spined stickleback fish. A sexually mature stickleback fish male will have a red-colored belly to attract the female. A mature stickleback fish migrates to warm shallow waters. Migration is an instinctive behavior triggered by the increase in gonadotropic hormones, which is in turn controlled by environmental factors like temperature and photoperiod. On reaching shallow waters, the fish builds a nest, which is open at both ends, with the help of plants and weeds. The nest-building behavior is triggered by the presence of green vegetation in the water. Thus he acquires a small territory. He exhibits territoriality or defends the area around the nest from other male fishes that are also in search of suitable nesting grounds. The fish becomes aggressive and exhibits threat posture by facing downwards when another male tries to invade territory. The sign stimulus for territorial and aggressive behavior is the red belly of other male fish. In an experiment where males were displayed with realistic model without red belly and red bellies models of variable shapes, Timbergen observed that males did not respond to realistic model that lack red bellies, but various shape red-bellied models could stimulate the response just as well as real makes, supporting sign stimulus hypothesis. Figure 92. The presence of a female triggers the courtship behavior in the male because of its swollen belly that is full of eggs. The male performs a zigzag dance which attracts the female. During his dance, the male always exposes his red belly to the female. The male leads the female into the nest to releases its ova that are immediately fertilized by the male. After this the female is driven out and the male may lure other females to its nest. The presence of eggs in the nest triggers parental care in the male fishes and begins to fan the eggs with its fins, and young ones are taken care by the males. Hence various stereotyped instinctive behavior in stickleback fishes are migration, nest building, territoriality, aggression, courtship and parental care. Timbergen conducted simple experiments to demonstrate the instinctive behavior pattern in stickleback fish. Males presented with models of stuffed female, or even a bloated male elicited the courtship dancing behavior. When a female is presented with a red belly model of the male fish, it faithfully followed it, suggesting the visual sign triggered these behaviors. Example 3. Food begging behavior in herring gull. Food begging is common instinctive behavior of hunger birds. Young birds beg for food. from their parents by opening their mouth wide and making chirping sound to attract the parents' attention. This seems to elicit the instinctive parental behavior in the parents, and they will feed the hungry mouths of their offsprings. Tinbergen and A.C. Predeck carefully examined the releasers involved in the interactions between herring gulls and their chicks during feeding. An adult herring gull has a red dot at the end of its bill, figure 93. When the gull returns to its nest with food in its stomach, its chicks beg for food by pecking at the red dot, thereby stimulating the adult to regurgitate the food for the chicks to eat. Tinbergen and Predeck hypothesized that the red dot was a releaser for the chicks' begging behavior. To test their hypothesis, Tinbergen and Predeck made paper cutout models of gull heads and bills, varying the colors and the shapes. Then they rated each model according to how many begging pecks it received from naive, newly hatched gull chicks, see figure 93. The shape or color of the model head made no difference. In fact, a head was not even necessary. The chicks responded just as well to models of bills alone as long as they included the red dot. The shape of the bill was important, however. A model with a long, thin bill elicited stronger responses than a realistic model. Clearly, the chicks had inherited the ability to recognize a simple set of stimuli and respond to them with their also inherited begging behavior. To the ethologists, this finding represented an example of a behavior that was genetically determined rather than learned. Figure 93, releasing the bagging response. Example 4, eggshell removal from nest by gulls. After gull nestlings hatch, the parents remove the eggshells from the nest. The eggshell removal is considered an anti-predation behavior, and is performed very soon after a chick hatches. The nest with eggshell can be easily detected by predator and thus more chance of nestling predation. Common black-headed gulls normally remove the eggshells within a couple of hours after chicks hatch, and sometimes within minutes. The shorebirds must remove empty eggshells and return to their nests quickly to protect their chicks from predators. There is no time for elaborate neural processing to operate. Research has shown that the first time breeders will remove the empty shells placed in their nests even before they lay their first egg. This suggests that the eggshell removing behavior is programmed in their genes. It is not a learned behavior. Tinbergen experimentally found that color is a crucial stimulus for the eggshell removal behavior. In particular, the common black-headed gulls tended to remove orange-colored eggshell models the most, among other unnatural eggshell colors such as blue and green. Also, the birds tended to remove eggs that appeared similar to their own white inner lining. Weight of egg can be another stimulus, as eggshells are lighter in weight as compared to intact eggs. Example 5, escape reactions of ducklings and goslings. Unlike the above examples, where a red belly or just a spot could generate an instinctive behavior, in this case, the whole or entire shape of the model was responsible for the V reaction. The ducklings and goslings in the farmyard show two different responses to the same model when it is sailed above them. If the model is sailed to the left, the young birds do not show any signs of running away. However, when the model was sailed to the right, they responded by running away. This escape response exhibited by the ducklings and goslings was due to fact that the models appeared differently when sailed in the opposite direction. When the model moved to left, the model appeared like a duck or goose, however when it moved to the right, it appeared as a predatory bird, like hawk or eagle, which elicited the fear response, which generates the escape reaction in the young ones. Example 6, shoving of owner's egg out of nest by cuckoo chick. In Europe, a parasitic bird called the common cuckoo uses a sneaky strategy to raise its babies, termed as brood parasites. It is trick another bird, fish or other animal into taking care of their young. First, a female cuckoo finds a nest built by a bird of a different species. For example, it might be a great reed warbler. Then, she sneaks into the warbler's nest, lays an egg and flies away. The warblers often accept the new egg. Indeed, they take care of it along with their own eggs. The cuckoo chick hatches before the warbler chicks. And it wants all the food from the warbler parents for itself. So the young cuckoo chick shoves the nest owner's eggs out of the nest. This is example of innate behavior, as there is no room for learning. The warbler parents keep feeding the cuckoo chick even as their own offspring are gone. 1.4 Conditions favoring innate behaviors. The ability to learn and to modify behavior as a result of experience is often highly adaptive. But there are many conditions where genetically determined behavior are more adaptive. 1.As we have seen in spiders, if role models and opportunities to learn are not available as in species with non-overlapping generations, then there is no alternative to inherited behavior. 2. Inherited behavior is also adaptive when mistakes are costly or dangerous. Mating with a member of the wrong species is a costly mistake. Thus the function of much courtship behavior, such as that of dabbling ducks, is to guarantee species recognition. In an environment in which incorrect as well as correct models exist, learning the wrong pattern of courtship behavior would be possible. 3. Inheritance of behavior patterns used to avoid predators or capture dangerous prey is also adaptive. These situations allow no room for mistakes. If the behavior is not performed promptly and accurately the first time, there may not be a second chance. 21.5 Experiments to distinguish innate or learned behavior. One goal of behavioral biology is to distinguish the innate behaviors, which have a strong genetic component and are largely independent of environmental influences, from the learned behaviors, which result from environmental conditioning. 2 Experimental approaches have been used extensively by ethologists to determine whether behavior is inherited. 1.5.1 Deprivation experiment. In a deprivation experiment, an animal is reared so that it is deprived of all experience relevant to the behavior under study. If it still exhibits the behavior is presumed to be inherited. In one such experiment, a tree squirrel was reared in isolation, on a liquid diet, and in a cage without soil or other particulate matter. When the young squirrel was given a nut, it put the nut in its mouth and ran around the cage. Eventually, it made stereotypic digging movements in the corner of its cage, placed the nut in the imaginary hole, went through the motions of refilling the hole, and ended by tamping the non-existent soil with its nose. The squirrel had never handled a food object and had never experienced soil, yet the stereotypic behavior of a squirrel burying a nut was fully expressed. If a behavior is not expressed during a deprivation experiment, it may nonetheless have genetic determinants. The right conditions may not have been available to stimulate the behavior during the experiment. The squirrel described above, for example, had to be given a nut for its digging and burying behaviors to be triggered. Specific stimuli are required to elicit the expression of many inherited behaviors. Konrad Lorenz and Nico Tinbergen conducted classic studies of the nature of the stimuli that elicit such behaviors and called such stimuli releasers. 1.5.2 Hybridization experiment. In a hybridization experiment, closely related species are interbred and the behavior of their offspring is observed. Many closely related species can be interbred, thus, it is possible to see whether their offspring have inherited elements of the behavior of one or both parents. Konrad Lorenz conducted hybridization experiments on ducks to investigate the hereditary basis of their elaborate courtship displays. Dabbling duck species such as mallards, teals, pintails, and gadwalls are closely related to one another and can interbreed, but because of the specificity of their courtship displays, they rarely do so in nature. Each male duck performs a carefully choreographed water ballet that is typical of his species, figure 94. A female is not likely to accept his advances unless the entire display is successfully and correctly completed. When Lorenz crossbred dabbling duck species, he found that the hybrid offspring expressed some elements of the courtship displays of each parent species, but expressed them in new combinations. Of particular interest was his observation that the hybrids sometimes showed display components that were not in the repertoire of either parent species, but were characteristic of the displays of other species. Lorenz's hybridization studies clearly demonstrated that the stereotypic motor patterns of the courtship displays were inherited. The observation that females were not interested in males performing hybrid displays was evidence that natural selection was shaping these genetically determined behaviors. 1.6 Learning. New generations of behavioral biologists have moved beyond the ethologists' focus on inherited behavior to show that most behavior involves an interaction between inheritance and learning. The begging behavior of gull chicks is an example of such an interaction. Although newly hatched chicks respond maximally to simplistic artificial releasers, they gradually learn to discriminate between models and real gull heads, and they eventually beg only from their own parents. Thus, the inherited ability to recognize a simple releaser is subsequently refined by learning. Thorpe defined learning as a long-lasting adaptive change in behavior resulting from past experience, hence it is acquired during the lifetime of an individual. Learning is a relatively permanent change in behavior as a result of experience. It is a process by which animals modify their behavior, or adapt to their environment, in ways that allow them to experience increased fitness. The early ethologists did not ignore learning or deny that it took place. In fact, they pioneered the study of learning. Tinbergen performed an early study of spatial learning, by which an animal learns to recognize features in its environment. In a classic experiment, he placed objects such as pine cones near the entrance of a nest dug by a female digger wasp. After the wasp left her nest, he moved the objects a short distance away. Upon returning, the wasp oriented to the moved objects and could not find her nest entrance. She had learned to recognize objects in the environment to use as orientation cues. 11.7 Characteristics of learned behavior. 1. It is acquired during the life of an organism due to constant experience. 2. It is experience-dependent and can be modified through experience. 3. Learning is flexible. 4. Learning behavior differs from individual to individual among the same species, hence not species-specific. 5. Learned behavior is certainly not inherited, though the ability to learn is almost certainly inherited, as it is dependent on the development of the nervous system of the organism which is inherited. 6. All organisms from protozoa to humans have the ability to learn at least to some extent. 7. However, learning in humans surpasses all other animals. Almost everything we do has been learned. No other species surpasses the humans in their amount and range of information that is acquired through learning. 21.8 Types of learning. Following types of learning have been identified among animals. 1.8.1 Habituation. Habituation is the simplest and most common type of learning seen in all organisms, as simple as protozoan to as complex as man. It can be defined as the gradual decrease in response to repeated exposure to the same stimulus if found to be harmless. Hence it is a kind of negative learning wherein the animal learns to ignore a harmless stimulus and fails to respond. Thus habituation is the ability to ignore a prolonged stimulus. It is the gradual learning by an organism not to respond to stimuli which are repeatedly found to be of not adaptive value to it. Advantages. 1. It saves energy by avoiding unnecessary response. It is a vital process because without it, the animals bombarded by the numerous visual, auditory, olfactory, gustatory, and tactile stimuli from their environment would be in a constant and needless state of alarm or expectation. 2. Habituation behavior filters out the multitude of background stimuli that have no important consequences, thus leaves the animals attention free to concentrate on the stimuli which are essential for its survival and which may be potentially harmful. Example 1. If a farmer introduces a scarecrow in his field, initially crows will tend to flee away from scarecrow, but after time, birds learn to ignore scarecrows as they will learn that it is harmless. Figure 95. Figure 95, Crows get habituated by scarecrow, kept for prolonged time. Example 2. If the web of a spider is disturbed at one end, but finding nothing, it returns. If this stimulus is repeated severe times, it will gradually stop responding, as the stimulus was of no important consequence to the spider, and hence it will ignore the stimulus. However, the spider will respond if some other end of the web is disturbed. Example 3. Prairie dogs give alarm calls when mammals, large birds, or snakes approach, which warns others of the presence of these predators. These alarm calls facilitate escape into their burrows. However, when their burrows are located near trails used by humans, gives alarm calls every time a person walks by would be a wasted effort. Thus, habituation to humans is an important adaptation to these animals. 1.8.2 Imprinting. Imprinting is a relatively simple but specialized type of learning that takes place during critical period or sensitive period early in the life of an organism, for a complex set of stimuli that can later serve as a releaser. It is a especially rapid and relatively irreversible learning process that occurs early in life. The central concept of imprinting is the innate release mechanism whereby organisms genetically predisposed to be especially response to certain stimuli during the critical period. The classic example is the imprinting of offspring on their parents and parents on their offspring to ensure individual recognition even in a crowded situation such as a colony or a herd. It was first described by O. Heinroth and later demonstrated by Konrad Z. Lorenz on goslings and ducklings, which is also called following behavior. Lorenz hatched goose eggs in an incubator, and because he was the first thing the goslings saw when they hatched, they imprinted on him. The goslings followed him everywhere, interacting with him as if he were their parent. When the experiment was repeated by his assistants, each wearing boots with a different design, the goslings imprinted on the boots, and would follow only a person wearing the boots they first saw when they hatched. Characteristics of imprinting. 1. Imprinting will occur only during the critical period of an animal's life. The critical period for imprinting is determined by a developmental or hormonal state and can be quite brief. If a mother goat, for example, does not puzzle and lick her newborn within 5 to 10 minutes after its birth, she will not recognize it as her own later. In this case, imprinting depends on olfactory cues, and the critical period is determined by the high levels of the hormone oxytocin in the mother's circulatory system at the time of birth. The time of the critical period varies and it takes place from 13 to 16 hours of hatching, and no imprinting occurs after 36 hours. Human babies have a sensitive period that occurs between 18 months to 3 years. During this critical period, the absence of the mother or a foster caretaker results in behavioral abnormalities. Later in life, they are unable to show normal bonding. 2. Imprinting is a form of learning behavior where the young ones learn to recognize an object or person considering it to be their parent. Hence develop an almost irreversible attachment to it. Thus, imprinting is an innate behavior, but the recognition of the object is learned. It is innate as it cannot occur any time during the life of an animal, but it is seen only during a particular period called critical or sensitive period. 3. Imprinting occurs towards any moving object or person first seen by the animal. It was first seen in birds, but later also observed in fishes and mammals. 4. Imprinting is relatively permanent. Advantages of imprinting. 1. Though imprinting is seen for a short period in the years, there is a long-lasting effect. 2. Imprinting influences social behavior and mate selection. 3. It allows the young ones to recognize their own kind and especially their parents, which is necessary for their survival. 1.8.3 Associative learning. Associative learning simply means that an animal learns to associate an event with a result. There are 2 types of associative learning: classical, Pavlovian, conditioning, and instrumental, operant or trial and error, conditioning. 1. Classical conditioning, or Pavlovian conditioning. A type of learning in which a novel stimulus is paired with an existing stimulus and elicits a particular innate response. Eventually, the novel stimulus alone elicits the same response as the existing stimulus. Classical conditioning begins with an innate response to a stimulus, such as salivating in response to the sight of food. Pavlov called the food the unconditional stimulus, US, and salivating the unconditional response, UR. Next, Pavlov paired a neutral stimulus, such as the ringing of a bell, with the presentation of food. Such conditioning was done repeatedly until the animal learned to associate the bell with the arrival of food, figure 96. In Pavlov's terms, the bell was a conditional stimulus, CS, and salivation in response to the bell was a conditional response, CR. Subsequently, a dog would salivate after simply hearing the bell, even in the absence of food. A new predictable relationship had developed between a stimulus, bell, and a response, salivating. Often the word conditioned is used instead of conditional when referring to a stimulus or response. 2. Operant conditioning, trial and error learning. Operant conditioning or trial and error learning is a type of associative learning where particular actions can be reinforced by providing a reward after successfully completing the task. Thus, operant conditioning involves learning associations between learned behaviors and outcomes. Edward Thorndike and B.F. Skinner pioneered the study of operant conditioning, also known as instrumental conditioning. From Skinner's perspective, much behavior is learned from the consequences of this conditioning. Any of 4 types of operant conditioning can occur, depending on whether a behavior becomes more or less likely as a result of the presentation or removal of a stimulus, as follows. 1. In positive reinforcement, a behavior becomes more likely due to the presentation of a stimulus, such as food. 2. In negative reinforcement, a behavior becomes more likely due to the removal of a stimulus, such as pain. 3. In positive punishment, a behavior becomes less likely due to the presentation of a stimulus. 4. In negative punishment, a behavior becomes less likely due to the removal of a stimulus. Edward Lee Thorndike was one of the most famous and popular behaviorists of the 20th century who investigated this type of learning in cats using a series of puzzle boxes or problem boxes. The doors of these puzzle boxes could be opened by pushing a lever or button, pulling a string. The cat, confined in such a puzzle box, tries to escape by moving about randomly or restlessly inside the cage, and in this process accidentally it pushed the lever or pulled the string and the door opened, which resulted in obtaining its food. This process was repeated for the next few times till the cat learned to associate the pushing of the lever or pulling of the string resulted in obtaining the reward i.e., food. Figure 97. Gradually, after several attempts or trial and errors, the cat learned to eliminate all activities that did not get any reward, and at the same time it reinforced or repeated the activities that were rewarding. Finally, the cat learned to push the lever or button or pull the string to open the door as soon as it was confined in the box without making any errors. Figure 97, Thorndike's puzzle box. Cats solve problems solely by trial and error method. B.F. Skinner, another famous American behaviorist, performed a number of operant or trial and error learning experiments in his famous Skinner box by using model animals like rats and pigeons. The Skinner box experiments were slightly more complex as it had more variables introduced in the experiment. The Skinner box had a red light, green light, loudspeaker, a lever, a food dispenser, and the floor had an electric grid. The rat in the box learned through trial and error that it could obtain the reward, i.e., food, when it pressed the lever only when the green light was switched on, while pressing the lever when the red light was switched on did not fetch any reward, and pressing the lever when loud speaker was on resulted in an electric shock. Figure 98. Figure 98, Skinner box and operant conditioning chamber. The rat gradually learned to press the lever to obtain the reward or food when the green light was on. It gradually avoided the activity of pressing the lever when red light was on, as it did not get any reward. The number of trials and errors made to associate loud sound with the Electric shock or the punishment were much fewer when compared to the number of trial and errors made to learn to associate red light with no reward. This type of learning where the animals learn to repeat or reinforce activities that are pleasurable or fetch a reward and avoid activities that are painful or associated with punishment is called trial and error learning or instrumental or operant conditioning. Another simple experiment conducted by Skinner to demonstrate trial and error learning was a T-shaped maze where the animal must turn to the right or left, figure 99. On one turn the animal may find its reward, i.e., food or escape, while on the other turn it receives a punishment, i.e., an electric shock. By series of trials, some successful and others unsuccessful, the animals learn to take the favorable route, which is rewarding, and avoid the unfavorable route, and its performance improved with time. Ultimately, the animal learns to make the correct choice almost all the time. Learning through random and often erratic activity is called trial and error learning. The animal's capacity to learn is indicated by the number of trials required before the animal makes the right choice. Figure 99, T-maze for operant conditioning. Thus in trial and error learning, an animal faced with several alternatives is rewarded, positively reinforced, for making the proper choice and learns to make this response without hesitation. This type of learning is employed by animal trainers to make animals learn all types of tricks by using positive reinforcement. Skinner believed that humans too are controlled primarily by positive reinforcement, although many disagree. This idea. 1.8.4 Latent or exploratory learning. Latent learning, sometimes called exploratory learning, involves making associations without immediate reinforcement or reward. The reward is not obvious. An animal is apparently motivated, however, to learn about its surroundings. For example, if a rat is placed in a maze that has no food or reward, it explores the maze, although rather slowly. If food or another reward is provided, the rat quickly runs the maze. Apparently, previous learning of the maze had occurred but remained latent or hidden until an obvious reinforcement was provided. Latent learning allows an animal to learn about its surroundings as it explores. Knowledge about an animal's home area may be important for its survival, perhaps enabling it to escape from a predator or capture prey. Example, in mice, knowledge of the immediate environment of its burrow may help it escape from a predator. At the time of acquiring this knowledge, it has no apparent value, hence not all behavioral activities are apparently directed to satisfying a need or obtaining a reward. 1.8.5 Social learning. In social species, other individuals are a source of information for learning, a phenomenon known as social learning. In individual learning, such as trial and error learning, information is acquired through an individual's own activities, while in social learning, individuals learn by observing others. Social learning can be favored by selection when it reduces the time and energy costs of learning. Young animals have much to learn about their environment, and for many species, one of the most important lessons is how to avoid being eaten. 1. Local enhancement. The juveniles, young prairie dogs reared with experienced adults, had higher survivorship. Juveniles learn anti-predator behaviors from experienced adults, and that learning helps them survive. For example, birds such as terns feed on fish near the water's surface by diving into the water. Their food patches, schools of fish, are always moving, which can make locating food especially challenging. Individuals will often use a strategy known as local enhancement, in which they look to other foraging individuals as a cue to the location of food. This phenomenon is an important aspect of social learning. 2. Public information. It is the information obtained from the activity or performance of others about the quality of an environmental parameter or resource. 4. Example, sticklebacks fish also use public information to estimate V-patch quality. Sticklebacks were apparently uses the number of fish at each food patch is a cue to its quality. Even experimentally, the site where more number of fish model are kept may not have any food at all. 3. Teaching. The hallmark of teaching is the active participation of an experienced individual in facilitating learning by a naive conspecific. Animal teaching has been defined in many ways, but 3 widely accepted criteria for evidence of teaching are: 1.1 Experienced individual. The teacher modifies its behavior only while in the presence of a naive pupil. 2. The behavior is costly for the teacher, and 3. The pupil acquires knowledge or a skill more rapidly through the actions of the teacher than it would otherwise. Example 1. Ptarmigan hens teach chicks their diet. Ptarmigans feed on a variety of leaves, flowers and buds, and berries of shrubs and wildflowers. Their natural habitat contains many dozens of plants, but the diet of ptarmigans is composed of just a small subset of available plants. Chicks follow hens for several weeks after hatching. Occasionally, when one or more chicks are nearby, a hen will drop bits of food it is eating, produce a unique vocalization, a food call, and bob its head up and down. In response to this food call display, nearby chicks will run to the hen and feed. These displays a form of teaching. Food call displays always resulted in chicks feeding on the plant the hen had been feeding on, and there was a positive correlation between the proportion of food calls associated with those species and the proportion of food in the diet of the chicks. Chicks of hens that produced the greatest number of food call displays consumed significantly more protein in their diet than chicks of hens that produced the fewest food call displays. Clearly, hens were biasing food call production to plants high in protein, and chicks learned to consume a diet high in protein. These data are consistent with the definition of teaching given above, although the cost of teaching to the parent was only inferred. A. Reduced feeding rate. Example 2, tandem running in ants. Temnothorax albipennis, an ant common throughout Europe, lives in small colonies that often contain fewer than 100 workers. Colonies are located in loose, rubble or small rock crevices, and when these sites become unsuitable, colonies often move to more stable, higher quality locations. A small number of scouts are always searching the environment for food or better nest locations. When a valuable resource is located, the scout returns to the nest to recruit additional workers. Recruitment takes the form of a tandem run, 2 individuals moving together, in which the experienced scout physically teaches the naive worker to the resource. Franks and Richardson, 2006, videotaped the movements of 8 independent tandem runs, including the movement of the first scout to find the food, its behavior while engaged in tandem running with a naive recruit, and the movement of the recruit back to the nest. Frame-by-frame analysis indicated that leaders moved toward the food when the follower maintained antennal contact by tapping on the legs and body of the leader. Occasionally, the recruit would break away from the leader and engage in a circuitous movement, perhaps acquiring visual information about the environment to learn the route it was traveling. When physical contact was broken, the leader would slow down or stop moving until the follower was again close enough to maintain contact. Slowing down to wait for the recruit meant that tandem runs were costly for the leader. It took 4 times longer for the leader to move between the nest and food when engaged in a tandem run. However, recruits engaged in a tandem run located the food significantly faster than naive scouts. They benefited from the behavior of the leader. Tandem running behavior satisfies all 3 primary criteria for teaching. Furthermore, leaders modify their behavior in response to the behavior of followers in such a way that facilitates learning, and the information learned by the followers allows them to later become tandem leaders/teachers themselves. This demonstrates that teaching behavior is not limited to vertebrates. 11.9 Innate and learning both can interact for development of behavior. Many behavior patterns result from intricate interactions of inheritance and learning. One example that has been the subject of some elegant experiments is bird song. Adult male songbirds use a species-specific song in territorial displays and courtship. For most species, such as the white-crowned sparrow, learning is an essential step in the acquisition of song. If the eggs of white-crowned sparrows are hatched in an incubator and the young male birds are reared in isolation, their adult songs will be unusual assemblages of sounds, not the typical species-specific song. White-crowned sparrows cannot express their species-specific song without imprinting on that song as nestlings, Figure 100, Experiment 1. But even though the male white-crowned sparrow must hear the song of his own species as a nestling to sing it as an adult, he does not sing it as a juvenile. Instead, the auditory imprinting he experiences as a nestling forms a song memory in his nervous system. As the young male sparrow approaches sexual maturity the following spring, he tries to sing, and eventually he matches his imprinted song memory through trial and error. If a bird that has heard his species-specific song as a nestling is deafened before he begins to express his song, he will not develop his species-specific song, Figure 98, Experiment 2. The bird must be able to hear himself to match his song memory. If he is deafened after he expresses his correct species-specific song, however, he will continue to sing like a normal bird. 2 periods of learning are essential, the first in the nestling stage, the second as the bird approaches sexual maturity. Researchers first became aware of the importance of social factors in bird song learning when they discovered that individuals of some species will not learn from taped song, but instead require live birds as their song tutors. The sensitive imprinting period for white-crowned sparrows closes at approximately 50 days, and that songs of other species presented during the sensitive period are uniformly rejected. When the same bird become adult and tutor is present, it tries to sing, but the song is totally different from what it heard during imprinting, which was rejected, from another species as well as what is performed by other member of same species. Adult bird cannot learn song in crystallized way when the space-specific song was presented during imprinting. 11.10 Hormones and behavior. All behavior depends on the nervous system for initiation, coordination, and execution. Frequently, however, it is the endocrine system, through its controlling influences on the development and the physiological state of the animal, that determines when a particular behavior is performed, and even when certain behaviors can be learned. We will discuss 2 complex cases in which hormones control the development, learning, and expression of behavior, the development and expression of sexual behavior in rats, and the maturation of the brain regions required for song learning and expression in birds. 11.10.1 Sex steroids and sexual behavior in rats. Differences in the behavior of males and females of a species are clear examples of genes influencing the development and expression of behavior. Such sex differences in behavior are the result of actions of the sex steroids on the brain. Rats, like most other animals, have stereotypic sexual behaviors. A female rat in estrus, a period of sexual receptivity, responds to tactile stimulation of her hindquarters by assuming a mating posture called lordosis. A male rat, encountering a female in estrus, engages in stereotypic copulatory behavior. The roles of genes and sex steroids in the development and expression of lordosis and male copulatory behavior have been investigated through experiments that manipulated the exposure of the newborn and adult rat brain to sex steroids. Experiments such as those shown in Figure 101 led to 3 conclusions. Development of male sexual behavior requires the brain of the newborn rat to be exposed to testosterone, but development of female sexual behavior does not require the neonatal brain to be exposed to estradiol. Exposure of newborn rats to testosterone masculinizes the nervous systems of both genetic males and females so that they express male sexual behavior as adults. Sex steroids are necessary for adult rats to express sexual behavior. Moreover, the male sex steroid, testosterone, has an effect only in males, and the female sex steroid, estradiol, has an effect only in females. Thus, the sex steroids that are present during development determine which pattern of sexual behavior develops, and the sex steroids that are present in adulthood determine whether that pattern is expressed. 11.10.2 Testosterone and songging in birds. We have already seen that learning is essential for the acquisition of bird song. Both male and female songbirds hear their species-specific song as nestlings, but only the males of most species sing as adults. Male birds use song to claim and advertise territory, compete with other males, and declare dominance. They also use song to attract females, which suggests that the females know the song of their species, even if they do not sing it. Do sex steroids control the learning and expression of song in male and female songbirds? After leaving the nest where they heard their father's song, young songbirds from temperate and Arctic habitats migrate and associate with other species in mixed-species flocks. During this time they do not sing, and they do not hear their species-specific song again until the following spring. As that spring approaches and the days become longer, the young male's testes begin to grow and mature. As his testosterone level rises, he begins to try to sing, matching his own vocalizations to his imprinted memory of his father's song. Even if he is isolated at this time from all other males of his species, his song will gradually improve until it is a proper rendition of his species-specific song. At that point, the song is crystallized. The bird expresses it in similar form every spring thereafter. Why don't the females of most songbird species sing? Can't they learn the patterns of their species-specific song? Do they lack the muscular or nervous system capabilities necessary to sing? Or do they simply lack the hormonal stimulus for developing the behavior? To answer these questions, investigators injected female songbirds with testosterone in the spring. In response to these injections, the females developed their species-specific song and sang just as the males did. Apparently females form a memory of their species-specific song when they are nestlings and have the capability to express it, but they normally lack the hormonal stimulation. What does testosterone do to the brain of the songbird? A remarkable discovery revealed that each spring, an increase in testosterone causes certain parts of the male's brain necessary for learning and expressing song to grow larger, Figure 102. Individual neurons in those regions of the brain increase in size and grow longer extensions, and the numbers of neurons in those regions increase. Such research on the neurobiology of bird song has revealed that hormones can control behavior by influencing brain structure as well as brain function, both developmentally and seasonally. 11.11 The genetics of behavior. To say that behavior is inherited does not mean that specific genes code for specific behaviors. Genes code for proteins, and there are many complex steps between the expression of a gene as a protein product and the expression of a behavior. In no case are all the steps between a gene and its influence on a behavior known. Nevertheless, it is clear that behavior has genetic determinants. We will focus on 3 approaches to investigating how genes affect behavior: hybridization, artificial selection and crossing of the selected strains, and molecular analysis of genes and gene products. 11.11.1 Hybridization experiments confirms genetically determined behaviors. The effects of hybridization on the courtship displays of dabbling duck species were the subject of a classic ethological experiment, as we saw above. A more recent set of hybridization experiments was performed on crickets. The songs of crickets, like bird songs, are species-specific, and as in birds, only male crickets sing. They do so by rubbing one wing against another wing that has a serrated edge. The sounds they produce can be recorded and analyzed quantitatively. When 2 species of crickets were crossed, their offspring, the F generation, expressed songs that had features of the songs of both parental species. Backcrosses of F individuals with the parental species produced individuals that had songs closer to that of the parental species used in the backcross. Clearly the genetic background determined the song pattern. What was amazing, however, was the demonstration that female preferences for male songs were under similar genetic control. Given a choice, females from each parental species preferred the calls of males from their own species, but hybrid females preferred the calls of hybrid males. These genetic differences between the two parental cricket species and the hybrids were reflected in the properties of their nervous systems. When specific neurons in the crickets' brains were stimulated, songs that reflected the genotypes of the crickets were expressed. v. 11.11.2 Artificial selection and crossbreeding experiments reveal the genetic complexity of behavior. Domesticated animals provide abundant evidence that artificial selection of mating pairs on the basis of their behavior can result in strains with distinct behavioral as well as anatomical characteristics. Among dogs, consider retrievers, pointers, and shepherds. Each has a particular behavioral tendency that can be honed to a fine degree by training. However, dogs and other large animals are not the best subjects for genetic studies. Most artificial selection experiments in behavioral genetics have been done on more convenient laboratory animals with short life cycles and large numbers of offspring, such as the fruit fly, Drosophila. Artificial selection has been successful in shaping a variety of behavior patterns in fruit flies, especially aspects of their courtship and mating behavior. Crossing of these artificially selected strains reveals that most of these behavioral differences are due to multiple genes that probably influence the behavior indirectly by altering general properties of the nervous system. Few behavioral genetic studies reveal simple Mendelian segregation of behavioral traits. An exception is nest cleaning behavior in honeybees. One genetic strain of honeybees practices nest cleaning, or hygienic, behavior, which makes them resistant to a bacterium that infects and kills the larvae of honeybees. When a larva dies, workers of this strain uncap its brood cell and remove the carcass from the hive. Another strain of honeybees does not show this hygienic behavior and therefore is more susceptible to the spread of the disease. Figure 103. When these two strains of honeybees were crossed, all members of the F generation were non-hygienic, indicating that the behavior is controlled by recessive alleles. Backcrossing the F with the hygienic parental strain produced the typical 3:1 ratio expected for a 2-gene trait in the F2 generation. The behavior of the non-hygienic F2 individuals was also interesting. 1/3 of them showed no hygienic behavior at all, 1/3 uncapped the cells of dead larvae, but did not remove them, and 1/3 did not uncap cells, but did remove carcasses if the cells were open. Even though these results suggest a gene for uncapping and a gene for removal, these behavior patterns are complex. They involve sensory mechanisms, orientation movements, and motor patterns, each of which depends on multiple properties of many cells. The genetic deficits of non-hygienic bees could influence very small, specific, yet critical properties of some cells. If even a single critical property, such as a crucial synapse or a particular sensory receptor, N were lacking, the whole behavior pattern would fail to be expressed. Thus there is no specific gene that codes for the entire behavior. 11.11.3 Molecular genetic techniques reveal specific genes that influence behavior. Molecular geneticists are investigating specific genes that influence behavior. Male courtship behavior in fruit flies is a subject of many such studies. This behavior is stereotypic, species-specific, and requires no learning. A male recognizes a potential mate, follows her, taps her body with his forelegs, extends and vibrates one wing, and licks the female's genitals. If the female is receptive, the male copulates with her. Research in molecular genetics has now shown that most of this male courtship behavior is controlled by a single gene. In fruit flies with two X chromosomes, females, a gene called sex-lethal, SXL, is expressed. This gene is at the top of a genetic hierarchy that determines all aspects of sexual differentiation and behavior in fruit flies. The SXL protein causes another gene, called transformer, TRA, to produce the female-specific TRA protein. Fruit flies without the TRA gene develop into males anatomically and behaviorally, regardless of how many X chromosomes they have. But it is still another gene in the sex determination hierarchy that is responsible for male behavior. In the absence of the TRA protein, two additional genes, called double sex, DSX, and fruitless, FRU, are expressed. The DSX gene controls the anatomical differentiation of males, and FRU causes the formation of a nervous system that expresses male courtship behavior. Mutations of the FRU gene do not affect male body form, but they disrupt male courtship behavior.